very practically, very, excuse me, all of these messages coming here, and who actually, was very, very instrumental in recruiting our first participants actually quite a few participants from a number of years. It's my great pleasure to introduce today's speaker, and this is Lisa James, my colleague. Lisa is the director of the Women's Healthy Brain Aging Project at the Brain Science Center at the Minneapolis VA Medical Center. She is adjunct associate professor of neuroscience and psychiatry at the University of Minnesota and a permanent staff member of the Brain Sciences Center and the Mental Health Service Line. She is the ideal clinical scientist, a clinical psychologist by training, and an accomplished brain scientist by post-training, and doing that for the past 10 years or so, with a good number of publications and important contributions to the field. So Lisa is directing the Women's Healthy Brain Aging Project, and most importantly, she's the holder of the Anita Kuning Endowed Chair in Women's Healthy Brain Imaging, which she represents very ably. So it's my pleasure to get the floor to Lisa, and looking forward for her lecture. Thank you very much, Lisa, and thanks to Melinda Bruski, our administrator, who really put together all of this meeting. Thanks again. Floor is yours, Lisa.
All right, thank you, Apostolos, and thank you for having me today to be the first lecturer of the annual lecture in honor of Lily. So today I'll be talking a little bit about the project that Lily helped conceive, the Women's Healthy Brain Aging Study. And this really is a legacy of Lily's life. So, so this is in fond memory of Lily. She was a compassionate clinician and a skilled researcher, first at Johns Hopkins and then here at the Minneapolis VA, where she was the director of the Women's Comprehensive Health Center here for quite some time. And during that time, in that role, she observed that she had several women that she met with who are quite advanced in age and had intact cognitive functioning. And this really spurred an interest in understanding what is happening in the brains of individuals as they age. There's such an emphasis on cognitive decline with age, but this really kind of turns that on its head and looks at, well, what is it that promotes healthy brain functioning? And so that led to the establishment of the Women's Healthy Brain Aging Study, which I have the honor of directing, and I'll be presenting this today. The study is, as I said, part of her legacy. So I don't have any conflicts of interest to disclose, and I just want to point out that the study sponsors had no role in the design of the study or any other aspects of the study. And the things I'm going to be talking about today do not represent the views of the US Department of Veterans Affairs or the US government. I want to start by setting the stage here by pointing out that our population is rapidly aging. So in 2015, about 19% of the US population, about 19% of the US population was age 65 and older, and that's expected to be 27% of the population in the United States by the year 2050. So the average US life expectancy has declined a little bit in the last couple of years due to COVID, but in general, for women, it's about 79 years, and men, 73 years. And so what that amounts to is as we age, that allows more time for insults and damage to accumulate. Now, there's all sorts of repair mechanisms in place to help us manage those insults and damage, but at some point, when those insults overwhelm the ability to repair, death is the outcome. And when we look at the leading causes of death in those 65 and older, the top 5, as of the most recent CDC report, is heart disease, cancer, COVID, stroke, and Alzheimer's disease. Now, what's remarkable about this is that in the last 15 years, death from heart disease has decreased by 11%. In the last 25 years, death from cancer has decreased by 25%. And since 2000, death from Alzheimer's has increased by 145%. So this amounts to 1 in 3 seniors is dying from Alzheimer's or a related form of dementia. Now you might think that if you live long enough, cognitive decline is probably inevitable, but in reality it's not. And we can start to glean some answers about whether or not that's inevitable by looking at areas of the world known as the blue zones. These are areas where people commonly live past the age of 100. Now in these areas, it's not just that they're living a long time, they're actually quite healthy. As well. So in these 5 regions, approximately 1/3 of the population lives to age 90, which is 2.5 times more than here in the United States. They have 20% less cancer, 50% less heart disease, and virtually no dementia, despite living to quite ripe ages. So I have this picture here in Apostolos. I apologize, I'm probably saying the name wrong, but I'm going to call it Ikaria. And they've dubbed this the island where people forget to die, because it's quite common for people to live well into 100 and older. Now, we're not in a blue zone, but we do have individuals in our community that are living to advanced stages and maintaining cognitive functioning. And that really brings us to our Women's Healthy Brain Aging study. So this study started in 2010. And as I mentioned, initiated by Lily's observation here at the Women's Veterans Clinic. I apologize. Sorry, getting calls on my work phone. My apologies for that. This study was funded by a group of women here in the Twin Cities, pictured here, Anita Kunin, Barbara Forster, and Sally Kling, who recognized the importance of studying brain health in women. And they actually raised the money to begin this research. And then subsequently established an endowment that's supporting the study. And Anita Kunin was sort of spearheaded that. So let's see. So we have Lily here, Dr. Georgopoulos, who's a world-renowned neuroscientist who really helped facilitate starting this study. I mentioned our funders, and then here's myself and some of my core staff that's involved in the study, our study coordinator and our lab personnel. Now, the overall goal of this study is to provide a comprehensive, multimodal, integrative assessment of brain status as it relates to cognition, genetics, and lifestyle across the lifespan. And the guiding questions for this study is, how does the brain change over the course of the lifespan to promote healthy functioning? And why are some brains more resilient than others? So I have this picture of Betty White here. I sort of think of her as an exemplar. She just recently passed away, as you may all recall, just prior to her 100th birthday. And up until her mid-90s, she was still very active in films, doing a lot of voice work in movies and things like that. So I sort of think of her in this regard, in terms of maintaining cognitive health across the lifespan. Now, the study aims, excuse me, there we go. The aims of the study are to characterize brain status with brain imaging and cognitive screening, and to associate that brain status with things like genetics and lifestyle factors like diet, exercise, and social connectedness. And I have genetics highlighted here because I'm really going to be emphasizing that primarily today. In addition, we want to assess the changes in brain status over time. So we have about 300 women participating in the study. Their ages range from 24 to 104, and they come in annually. So some of these women we've had come in for 10 years in a row now. So we can really track changes in their cognitive status and changes in brain function associated with that. In a sense, we can kind of think of this as the brain equivalent of the Framingham heart study, right? We know what a healthy heart should look like at various ages. The question, though, is what should a healthy brain look like at various ages? And ultimately, the goal is to use this data to forecast brain status. So can we look at somebody, say in their 40s or 50s, look at their brain function and predict whether or not they may ultimately have age-related cognitive disease? And then ultimately, ideally, can we intervene? So here we have this diagram that shows that as we age, our cognitive functions or health generally increase until we reach about middle age. And at that point, there's generally 3 trajectories. The bottom trajectory here is what's called cognitive aging. So in this trajectory, shortly after midlife, individuals might start experiencing cognitive decline. There's a relatively rapid drop off on that trajectory. Normal aging might have an extended period of time with sustained cognitive functioning before it drops off, and it doesn't drop off quite as steeply. And then there's this group of what's called successful agers. And these individuals, their cognitive functions maintain, they're relatively stable for an extended period of time. And when they do decline, they decline much less than those in the other trajectories. So ideally, we can identify people that are maybe at risk, intervene and bump them up to one of these other trajectories. Now, when we talk about genetics and cognitive function, we have to talk about apolipoprotein E. So ApoE is involved in lipid transport and metabolism, and it's involved in several central nervous system functions, including things like synaptic activity. And this gene exists in 3 isoforms. The E2 version, which is considered protective against Alzheimer's disease, the E3, which is neutral, and the E4, which is associated with risk for Alzheimer's. In fact, E4 is the strongest genetic risk for Alzheimer's disease, so much so that if you were to do genetic testing, such as through 23andMe, you can actually opt in whether or not you want to know if you carry this particular risk gene. And this risk occurs in a dose-dependent manner, such that if you do not carry the E4 allele, if you're a non-carrier, the frequency of Alzheimer's disease is about 20% in non-carriers, and the age of onset would be 84 years old. For those that carry one of these E4 risk genes, the frequency of Alzheimer's is 47%, and the age of onset is a bit younger, about 76. And for those that carry 2 of these alleles, there's a pretty high frequency of Alzheimer's in that population, and the clinical onset is younger, about 68. Now, what's interesting is that these particular isoforms, the primary difference is really a single amino acid change at 2 positions. So whether or not these isoforms carry what's called an arginine or a cysteine. So the E2, which is protective, has 2 cysteines, the E3 has 1, and the E4 has 0. And these amino acid changes really change the structure and the function of these isoforms. Now, typically people look at this in a dichotomous way, either presence or absence of a particular allele, like the E4. But we inherit 2 of these in a Mendelian fashion, and that results in 6 possible genotypes, 3 that are homozygous listed here, and 3 that are heterozygous. And what's interesting is that we can transform these genotypes into a continuous measure related to the number of cysteine residues per mole. So for instance, as it says here, the E2 would have 4. And then as we move down, the E4 has 0. Now, we were interested in looking first at how these cysteine residues per mole, which again influence the structure and function of these alleles, how they're associated with neural communication. So, the brain is this massively interconnected network of billions of neurons, and communication amongst those neurons is really the essence of brain function. And we can evaluate that brain function with the highest fidelity using magnetoencephalography, a type of neuroimaging that we utilize here in our research. And so here specifically, we looked at what we have termed the synchronous neural interactions, which is the cross-correlated neural activity recorded simultaneously from 248 meg signals. So this is really giving us an estimate of neural communication throughout the brain, because we're looking at 30,628 neural interactions across the brain. And what we found here is that there was systematic association between the number of cysteine residues per mole and neural communication. So I've shown here specifically that the variance in the synchronous neural interactions decreases with increasing number of cysteine residues per mole. And this is important because previous work in our lab had established that in general, healthy brains are very consistent in terms of their patterns of neural communication. So much so that variations in those patterns can actually be used to distinguish different disease states, which we've dubbed neural signatures. So we've identified several neural signatures of disease based on variation from what's considered normal or healthy. Now, we looked at the properties of these neural interactions. And specifically here, we looked at the flexibility of the neural interactions and the variability. So, the idea being that we want to have a flexible network. We've shown in other research that when the network is fixed, that is associated with various types of pathologies, specifically post-traumatic stress disorder, for instance, is associated with a fixed network. So here on the left panel, we show the what's called the covariance, absolute value of the covariance, and we've demonstrated that the genotypes that are associated with a greater number of cysteine residues per mole are more flexible. So here, the lower covariance reflects flexibility. And on the right-hand side, we have a different measure of network variability. And here again, we show that as the cysteine residues per mole increases, according to the genotypes listed here, again, we see greater network variability. Now, when we looked at network properties by decade and genotype, we found that they were relatively stable up until about the 7th decade. And then at that point, there's pretty significant divergence. Now, you might notice we don't have some of the genotypes listed here, and that's because the E2, and E4, are relatively rare, and we didn't have enough to look at several different decades. So we had to eliminate those here. But what we still see is that there's the same pattern, that the greater number of cysteine residues per mole continues to be associated with more flexibility and more variability in advanced stages. And in contrast, if we look at the top, the blue line, this has the fewest number of cysteine residues per mole of the groups that we established here. And after the 7th decade, that line sort of shoots up, both in terms of the covariance, so less flexibility, and the product of the standard deviation, which suggests greater variability. So essentially, this boils down to more cysteine residues per mole are associated with healthier network functioning. Now, we also wanted to look at how these network properties are related to cognitive performance. So women in our study completed the MoCA, which is a highly validated, well-established measure or index of, or screener rather, of cognitive functioning. And this is scored on a scale from 0 to 30. And a score of 26 or greater is considered evidence of normal brain functioning. Below that, we get into mild cognitive impairment, and then much lower is an indicator of likely Alzheimer's dementia. So all of these women here are scoring in the normal range, 26 or more. But it's interesting to note that as we approach that cutoff, the score of 26, we see a definite shift, right? A trend towards greater SNI, and greater variability. So stronger correlations and stronger variability. Okay, so more flexible and less variable networks are actually associated with better cognitive performance. So summarizing this first section, we've shown that ApoE genotype, and particularly the number of cysteine residues per mole, are associated with age-related changes in neural network flexibility and variability in cognitively healthy women, and that healthy cognitive functioning is associated with neural network flexibility and variability. And this is consistent with the prevailing literature that generally documents the relative benefit of ApoE2 over ApoE3, with ApoE4 being detrimental. And we're really, I think this is interesting in terms of sort of flipping the script, and instead of focusing less so on ApoE4, but really the benefit of ApoE2. But the bulk of the research really has been conducted on ApoE4. And a review summarized the numerous negative effects, loss of function associated with the presence of ApoE4, as well as the gain of toxic functions, including things like amyloid beta aggregation, tangle formation, but also aberrant brain activity and brain atrophy. And I have these circled because we're going to come back to these here in a few minutes. Now, this is, the ApoE story is really just a small part of the story. The bulk of the research that we've been doing in the last several years has been on a different set of genes, genes involved with the immune system, and in particular, the human leukocyte antigen. So the HLA region codes for cell surface glycoproteins that are involved in the human immune response to non-self antigens or foreign antigens. So this is the basis of how our body responds when we have an infection, say with viruses or bacteria. This is the basis of vaccinations working, of cancer neoantigens, tissue transplantation, and mate selection. So HLA is involved in a lot of critical aspects of maintaining health. Now, the overarching goal of HLA is host protection. So essentially, these are involved in detecting and eliminating foreign antigens, so things like viruses, bacteria, these cancer neoantigens. And there's two main classes of HLA that differ in terms of their location, their binding groove, source of antigens, and their general immune response. So we have class 1 HLA, which includes HLA A, B, and C. These are located on all nucleated cells. And they are able to, they have a relatively small binding groove, about 8 to 10 amino acid length. And they, the source of antigens there are endogenous or cytosolic antigens. And the immune response associated with class 1, it activates the CD8 cytotoxic T cells, which then promote cell death. For class 2, they are located on professional antigen presenting cells, so things like dendritic cells and macrophages. They're able to bind slightly longer amino acid length, about 10 to 30 amino acids, and they bind endocytosed exogenous antigens. And once they transport that to the cell surface, that activates the CD4 plus T helper T cells to activate B cells, which in turn creates antibodies and immune memory, so that if you're confronted with a particular antigen down the road, the body sort of kicks into gear and helps eliminate that. Now, this is the most highly polymorphic region in the entire human genome. There's actually over 30,000 different variations of this that have been identified and documented. And that polymorphism is really important at sort of the population level, because it really maximizes our protection against pathogens. That variation, although, which is most of the variation is in the binding groove, it contributes to variation disease protection and susceptibility. And this has been prominently documented for a number of autoimmune conditions, but it's increasingly documented for other conditions as well, both in our lab as well as many other labs as well. And one area that this is widely being investigated is in genome-wide association studies, where several GWAS studies have identified hits in this HLA region that are relevant to particular diseases. Now, everyone carries 12 of these, 6 class 1, 2 of each of the different genes there, and 6 of class 2. Now, this is important because the genes you have determine which antigens you can eliminate. So we have this little figure here that provides a little bit of a summary of what I've described already. So on the left-hand side, we have our class 1 HLA. And if we're exposed to a pathogen, if there is a match, if the HLA can bind with that pathogen, it will export it to the cell surface to present it to the CD8 T lymphocytes, which kills the cells and then eliminates that antigen or that pathogen. On the right side, we described a slightly different process where the class 2 molecules, if there is a match, will export that particular antigen to the surface, where it will activate the CD4 plus T cells, and that stimulates B cell activity or antibody production, again, eliminating the antigen and providing future protection through that immunological memory. Okay, so the key here is that the HLA matches, it can bind with a particular pathogen. But without a match, these antigens can persist. This entire process just kind of falls apart. And those persistent antigens can then contribute to damage. So when we think about the various roles HLA plays, there's several implications here. So if we have an infection, a virus or bacteria, and we have an HLA antigen mismatch, that could lead to, as we said, persistence of the antigen, and that might result in inflammation, autoimmunity, and ultimately disease. If we have a vaccination and we don't have a corresponding HLA, then that could result in reduced effectiveness of the vaccine, for instance, COVID vaccine, and in some cases might result in persistence of the vaccine antigen. And I just have here, for example, anthrax, which I will talk about very, very briefly here in a few minutes. And then with regard to cancer neoantigens, once cancer cells are, multiplying and mutating, they create, these neoantigens. And if we don't have HLA that is able to control those or respond to those, and the cancer continues to proliferate, and certain HLA has also been associated with poor treatment response. So several of these items, I have an asterisk here just to indicate these are all areas that we have published in the last several years. This last one, we haven't published in, but it's very relevant, which is tissue and bone marrow transplantation. This is the basis of things like Be the Match, which we have here locally. And another name for HLA is the major histocompatibility complex, because the HLA is really what determines whether or not tissue or bone marrow transplantations will be rejected. Now, because of this role in histocompatibility, large databases are maintained worldwide with HLA genotyping and for individuals to facilitate histocompatibility matching. And we have made use of those in some epidemiological data that I'll get to in a moment here. Now, I want to take a very quick detour and talk about something totally unrelated, but it was highly informative in terms of our healthy brain aging study, and that's Gulf War illness. So I'm here at the Minneapolis VA working with veterans. And Gulf War illness was a systemic illness that affected veterans of the 1990-1991 Persian Gulf War. And about 1/3 of US Gulf War veterans were affected. And individuals that have Gulf War illness report a lot of chronic diffuse symptoms that has sort of been of uncertain etiology. Historically. And the symptoms are things like fatigue, pain, neurological, cognitive, and mood symptoms, all of which involve the brain, as well as gastrointestinal, respiratory, and skin problems. These veterans tend to be in very poor health with high rates of physical and mental health conditions, and they have a lot of autoimmune disorders and other immune-related conditions, which suggest the possibility that HLA might be associated. So what we did is we first identified that amongst our Gulf War veterans, we had 144 unique alleles identified from those participants, and 6 of them, which all happened to be that class 2 HLA, they distinguished our Gulf War controls from those who had Gulf War illness. And there was a dose-dependent association with symptom severity. So you can see here that as individuals had increasingly more of these 6 protective alleles, the overall symptom severity significantly decreased. And the same was found for fatigue, pain, and neurological, cognitive, and mood symptoms. So again, as these individuals had more protective alleles, there was a reduction in these symptoms. Now, when we looked at the brains of individuals with Gulf War illness, we identified significant brain atrophy. So here we have an image of a veteran with Gulf War illness and an age and sex matched control Gulf War veteran. And you can clearly see significant atrophy. So the arrow there pointing at the cerebellum. And what this amounts to is about 12.5% atrophy per decade in the Gulf War veterans, compared to about 7.5% in the control Gulf War veterans. So accelerated brain atrophy here. Now, what's interesting is we looked at one of the alleles, one of the 6 that we had identified was protective against Gulf War illness. And we found that that particular allele, DRB1-1302, seemed to very clearly protect against that brain atrophy in Gulf War veterans. Now, this is interesting because this particular allele has been highly associated with protection against a number of conditions, primarily autoimmune conditions. And that got us thinking, well, this is, if it's protecting against atrophy, what about other conditions that are associated with brain atrophy, for instance, aging? So that actually, this is what started us investigating the role of HLA in brain aging. So here we looked at the effect of HLA on subcortical volume. We looked at cortical, subcortical, and total volume. And what we identified, what I'm showing here, is that in the individuals that did not have this protective allele, there was significant reduction in volume with age. On the right-hand side, though, we have the individuals that did have this protective allele, and there was no such effect. So this particular allele that was protective in Gulf War illness is also protected against age-related brain atrophy, and these findings were consistent regardless of the influence of ApoE. We looked at the influence of ApoE and did not change the effects here. Now, I'd also mentioned that earlier that ApoE is also associated with issues with brain function. And so here we looked at the presence or absence of this particular protective allele as it relates to brain function. So on the left-hand side here, we have individuals that have that E4 risk gene, and they did not have the protective DRB113 HLA gene. And what we see is with age, there's an increase in this network variability, which we've already established is detrimental. On the right side, though, now we have people that have the protective HLA gene and also have the E4 AD risk gene. And again, there is no such effect. So this risk gene is protective even in the presence of E4. Now, recently, we looked at the presence of this particular gene in relation to turnover in the bold signal. So this is just the sort of moment-to-moment change in the bold signal. And what we identified is that carriers of the DRB13 allele, which are shown here in blue, had significantly lower turnover, bold turnover, than non-carriers, which are shown in red. So this amounts to the systematic decreased hyperemia with DRB113. So the functional hyperemia is essentially the basis of the bold signal. And what we've proposed here is that individuals that have this protective allele are able to eliminate some sort of pathogen and then reduce the likelihood of having neuroinflammation that we might see that would lead to vasodilation, which we think we're seeing here in the non-carriers of this allele. Now, all of that I presented was on our healthy women, but we were interested in taking this a step further. And this is where that immunogenetics, population immunogenetics came into play, those large databases that are maintained all over the world in terms of HLA. So here we looked at the frequency of this gene that we identified in our sample of healthy women. We looked at that in 14 countries of continental Western Europe and looked at the association between that gene frequency and the prevalence of dementia. And as you can see here, there's a pretty clear association that as the frequency of this allele increases, it's higher in some countries than others, that the prevalence of dementia decreases. So this particular gene is protecting against dementia in Europe, and this is even after we account for the population prevalence of ApoE4, that risk gene. So this gene, which is related to immune system protection, against foreign antigens, it also protects against age-related changes in brain structure and function, and protects against dementia, even with this dementia risk gene. So that leads to other questions, like what about other protective HLA genes? We'd already established that HLA is the most highly polymorphic region of the human genome, so surely there's other genes out there. What is the mechanism? And then ultimately, can we intervene? So we went back to our population immunogenetic epidemiological research, and we identified 3 additional genes that are present in all 14 of the countries of continental Western Europe that were protective against dementia. And you can see these are all highly significant. Our favorite one here, the DRB113, has the steepest slope, indicating that it's quite protective, although it's less frequent in the population than some of these other genes. But when we sum these genes together, the frequency of these genes, we can see that about 80% of the population carries at least one of these alleles, and there's a robust association between the population prevalence of any of these alleles and the prevalence of dementia. So these genes are protecting against dementia. So at this point, we can kind of start working backwards in order to move forward, remembering that the goal of HLA is really about eliminating foreign antigens. So we can use bioinformatics to help us identify what antigens match HLA. Now, initially, just scanning a database referred to as the immune epitope database. This particular gene, DRB11302, had several hits with common viruses, things like alpha papillomavirus, which contributes to HPV, influenza, Hep B and C, etc. Now, what jumped out here is herpes viruses. And this is because herpes viruses have been widely implicated in Alzheimer's disease. Now, there are 9 types of herpes viruses that affect humans, and these result in common manifestations. So herpes simplex virus 1 and 2 result in oral lesions like fever blisters, cold sores, genital lesions. Other types like varicella zoster results in chickenpox and then subsequently shingles. And then others result in things like mononucleosis and roseola. These particular viruses are nearly ubiquitous, meaning just about everyone has been exposed to at least one of these viruses, if not several. And they're neurotropic, meaning they preferentially affect the central nervous system. And the way these work is that after initial infection, they establish lifelong latency and then are reactivated, particularly, for instance, under periods of stress, they can reactivate. And then when that reactivation happens, it can occur throughout the body. So the brain, the lungs, heart, kidney, and GI tract. So systemic effects can be seen. And the reactivation of these viruses has been associated with several clinical conditions. As I mentioned, Alzheimer's is key among those. Now, what's interesting is we looked at our 3 HLA alleles that we had established were protective against against dementia in continental Western Europe. And we compared the binding affinity of those particular alleles with other ones that are neutral with respect to dementia. And what we found is that the protective HLA alleles bind with significantly higher affinity. So a lower percentile rank here means better binding affinity. So the protective alleles are binding with higher affinity to all of the human herpes viruses. Excuse me. So that binding then means that they are able to ideally then eliminate those viruses. So we've proposed this mechanism that when we're presented with pathogens or harmful persistent antigens, if we have protective HLA, so DRB13, for instance, or other relevant HLA that we've established, everything's fine, right? Health ensues. But in the absence of those particular alleles, those pathogens and harmful persistent antigens may contribute to neuronal damage. And we believe that this is then where ApoE comes in. So we believe HLA is primary and ApoE is actually secondary to HLA. So that when neuronal damage occurs, ApoE swoops in. One of its roles is to facilitate neuronal repair. But the outcome of that is really dependent on one's HLA makeup. So if in the case of the ApoE2, which is protective against dementia, the ApoE3, which is neutral, neuronal repair ensues. But in the case of ApoE4, the ApoE4, due to those conformational changes that affect the structure and function, so those changes conferred by the absence of cysteine residues, they become these neurotoxic fragments that kind of just break off, and that ultimately contributes to additional neuronal damage. So the question then becomes, can we intervene? So we said, if someone has a pathogen and they don't have a matching HLA, that antigen might persist. And we're really looking at that as an intervention opportunity. So I want to go back briefly to some of the work that we did on Gulf War illness and kind of show you proof of principle about how this is working. So here we've taken serum from control veterans and veterans with Gulf War illness, and we've looked at 2 aspects in these cell cultures, spreading and apoptosis. So you can see in the first, the top left, the control serum, you see evidence of, lots of spreading and no evidence of apoptosis. The middle of that top panel is a veteran with Gulf War illness. And there we can see that the cells are actually clumping together. There isn't much spreading. And the red symbolizes the apoptosis there. Now, when we add serum from a Gulf War veteran, from a control veteran to that of the Gulf War veteran, that is actually correcting the issues that we're seeing in the Gulf War serum. So it's restoring the culture in terms of seeing spreading again and not seeing apoptosis. So that really suggests there's something in the blood of our control Gulf War veterans that's corrective here. So the next step, these bottom panels here, we assume there must be antibodies that the control veterans have that Gulf War veterans were not able to make, presumably due to not having compatible HLA. So here we added pooled antibodies, IgG, which is commercially available pooled antibodies. We added that to the cell cultures. And on the left, you can see the blue lines represent the Gulf War illness, and the red line represents the Gulf War illness with the addition of these pooled antibodies. And we can see that when we add these pooled antibodies, there's significant increase in spreading. Excuse me. And on the right-hand side, we can see that the addition of these antibodies results in a decrease of apoptosis. So really continues to speak to the role of antibodies and something lacking in the veterans with Gulf War illness. So then the next question is, can we then identify what is it in the serum of these Gulf War veterans? What sort of persistent antigens might be present, and can we remove them? So I mentioned briefly earlier the role of the anthrax vaccine. Gulf War veterans received a host of vaccines, one of which was the anthrax vaccine. And excuse me, we were able to identify by using specific antibodies that veterans, Gulf War veterans, had evidence of anthrax vaccine antigen PA63 in their serum. So we have here on the left, we have in red our Gulf War veterans, and the green are controls. And we see that the Gulf War veterans had a lot higher apoptosis. The middle panel shows that when we add the antibodies against the anthrax vaccine antigen, the red is the same as in the first panel, so Gulf War illness, but the blue now in the middle panel shows the addition of these antibodies, and we show a significant reduction in the apoptosis there. And the final panel is comparing the Gulf War veterans with the addition of the antibody against anthrax vaccine antigen to serum of controls. And we see that it's still has more apoptosis than controls, but significantly improved relative to what it was. So this was informative again in terms of the research now that we're doing with women. So now we have been collecting serum from women in our study and looking at evidence of apoptosis. And what we see here is that, again, there's this relative stability in terms of apoptosis until about the middle of the 7th decade, at which point there's almost this exponential increase in apoptosis. The other thing is that we're seeing evidence of apoptosis in some of the individuals that are a bit younger, so 40s and 50s. Now, when we look at the association between apoptosis and cognitive functioning, what we're seeing is that there's a robust association that with increased apoptosis, there's reduced performance on measures of cognitive functioning. So here we're looking at visual spatial scores on a cognitive screener that we're administering. So, what's ongoing now for us is the search for substances in the blood that might be adversely affecting the neural cell cultures, and that might contribute to cognitive decline. So can we identify what's happening, and then can we ultimately intervene? So summarizing this part, we've just shown how HLA is instrumental in foreign antigen elimination and also in maintaining brain health. So we have this schematic here where just a review that when we're exposed to a pathogen, if we have protective HLA, then that facilitates pathogen elimination and the consequence is no disease. However, in the absence of protective HLA, those pathogens might contribute to disease either through direct cell death or through resulting in chronic inflammation and in the presence of susceptibility, HLA, through autoimmunity, both of which might result in disease. So this really provides some novel opportunities for intervention along the lines of personalized precision medicine. So can we look at someone's serum, identify what's happening, ultimately remove it? Therefore promoting health in those individuals. So to summarize everything, back to our guiding question. So how does the brain change over the course of the lifespan to promote healthy functioning? Well, it seems that healthy brain functioning is associated with flexible or decorrelated networks and less variable networks. And that seems to be underlined in part by the number of cysteine residues per mole that each individual possesses based on their ApoE genotype. And why are some brains more resilient than others? Well, we think that having protective HLA or HLA that matches pathogens that you've been exposed to and or having limited exposure to those pathogens really promote resilience here. Also that I believe relates to the blue zones as well. That probably is part of the reason why these individuals, most of the blue zone communities are relatively isolated. And so they may either have limited exposure to outside pathogens or, the communities have generally have genetic protection against the pathogens that are present there. So I just want to point out as I'm wrapping up here that all of this work is from the Brain Sciences Center, which we are a multidisciplinary unit, and everyone that's listed here has contributed quite substantially. So we really approach brain health and disease from several fronts, including neuroscience, psychology, neurology, cell biology, physics, and bioinformatics. So none of this would have been possible without the contributions of all of these individuals, as well as our staff who have been integral in facilitating, actually acquiring the data and doing the lab work. So our study coordinator, Stacy Dolan, our lab personnel, Laura and Eric, and the computer science and MEG acquisition staff, so Matt and Dale, and our administrative assistant, Melinda, who's critical in maintaining the study. So that is all that I had for today. I want to thank you. I'm going to minimize my screen here. And I thought I had a pretty ambitious agenda, but we ended up getting through quite a bit or got through all of that rather quickly. So we do have a couple of minutes if anyone has any questions. Well, if there aren't any questions, if people end up having questions, you're more than welcome to contact me via email. You can reach me at lmjames@umn.edu.
Lisa, it does look like someone has their hand up.
Oh, okay, I did not see that.
Hi, Lisa. This is Gülin from the Minneapolis campus. Outstanding work and outstanding presentation. Thank you. I'm wondering if you have looked into long COVID and the HLA status and potential associations in that cohort at all.
Well, so yes, Dr. Georgopoulos and I actually just wrote a review looking at lung diseases in general and how we propose that all of them are linked to these persistent antigens. So long COVID was absolutely one of them. So yes, there's every reason to believe that HLA mismatch is associated both with limited vaccine effectiveness in individuals, with regard to COVID and other vaccines, but also long COVID. Yep, absolutely. Yeah, the other diseases that we looked at there, one was Gulf War illness, which I touched on today, but also chronic fatigue syndrome. So all three of those we believe are due to persistent antigens.
So I have a hand up. My name is Brown. I'm one of the participants, one of the subjects in your studies. And my question is, if you have the unfortunate experience of being short on this LHA, and thus at high risk for Alzheimer's, how much, if anything, can diet, exercise, and socialization, the other three major contributing factors, do to offset your risk?
Yeah, well, so HLA really plays a protective role. In my mind, I don't really think about HLA in terms of risk so much. But you're absolutely right that diet, exercise, social connectedness are very, very important. Particularly, especially if we look at, like, let's say the blue zones, for instance, right? They tend to often follow a Mediterranean diet. We know that anything that's good for the heart is also good for the brain. So if people are exercising, if they're eating, lots of fruits and vegetables, etc., that is going to be beneficial in terms of brain health. So absolutely, those modifiable risk factors are very, very important. Yeah, and I love that you mentioned the social part too, right? We often think about exercise and eating, but we know that social connection is actually very critical in terms of protecting against cognitive decline with aging. So yeah, all of these are very important.
So it might be able to delay the onset or whatever of something that you're highly at risk to get.
Yes, absolutely. Those modifiable factors play a huge role. And then the other thing I want to point out is that, I had mentioned we've identified 3 genes that were protective against the development of dementia in the broad population. We specifically looked only at genes that were present in all 14 of the countries that we were investigating. Now, remember, there are thousands and thousands of HLA genes, and they all are associated with dementia risk to varying degrees, either protective or susceptibility. So there's quite a few others that we didn't report in that particular study that are associated with protection from disease. Remember, the goal of HLA is host protection in general. So there are going to be a lot of these 30,000 variants that people have. A lot of them are going to be beneficial in terms of eliminating pathogens and protecting us. That's their very evolutionary goal. So there's more than just the one that we reported or the 3 that we identified. There's certainly many, many protective HLA genes. The main risk, the main genetic risk is that apolipoprotein E though. And in that case, those modifiable risk factors, lifestyle factors are very, very important. So,
thank you.
Yeah. All right, well, I'm mindful of the time. We're coming up to the hour here. So I just want to thank you all for attending. And again, I'm more than happy to take additional questions via email if anybody had any, but I really appreciate the time today. Thank you very much.
She's impressive.